Hi, my name is Hanna Choi. I'm a senior at UMass Amherst studying biology. And last semester, I had the great opportunity to learn under Dr. Lally at New England Retina Consultants as a researcher. And my poster is on a family of 4 members who came into the clinic with interesting retinal findings, and they were all carriers of the missense mutation in PRPH2. So the diseases we'll be talking about is CACD and pattern dystrophy. Both are inherited retinal diseases. CACD is typically found as unifocal atrophic lesions in the macula, meaning one large area of cell death in the macula. A great example is figure 1b. Pattern dystrophy, on the other hand, is typically found with a speckled pattern of drusen or lipofuscin. A great example is 4b. The effect on their vision is more severe for CACD in comparison to pattern dystrophy. Like I said, they're inherited retinal diseases, so they're associated with multiple genes, one being PRPH2. PRPH2 is a gene that encodes a protein that localizes to the outer segment discs of photoreceptor cells. So PRPH2 as a protein functions to stabilize these discs in photoreceptor cells to keep them alive and functioning. So to discuss about the patients, the proband initially came in to our clinic earlier last year, complaining of lowered visual acuity in her left eye, and we found unifocal atrophic lesions in both eyes. You can see that in figures 1b through E with large dots in the macula. We then tested her and her family of a 90-year-old mother and both her sisters, being 65 and 53 years old. The mother is figure 2, the older sister is figure 3, and the younger sister is figure 4. Dr. Lally diagnosed the proband, mother, and the elder sister's right eye to have CACD. The left eye of the elder sister and both eyes of the younger sister had pattern dystrophy. So right off the bat, you can see that there's phenotypic variability within one family. And we genetically tested all these members and found a missense mutation in the PRPH2 protein. And this change was from an arginine to a proline, which is pretty significant as it goes from polar to non-polar. So we suspect a change in the functioning of PRPH2. Additionally, the proband and both sisters were carriers of a PNPLA6 missense mutation, and PNPLA6 is a protein typically found in the axons of nerves, but can also be found in the photoreceptor cells. The proband and younger sister also carried a PROM1 missense mutation, and PROM1 is similar to PRPH2 as it is found in the outer segment discs of photoreceptor cells and is a protein that functions to stabilize it as well. The younger sister also carried an ABCA4 mutation, and she was heterozygous, which is interesting because homozygous ABCA4 missense mutations leads to Stargardt's. And literature said that ABCA4 can modify the phenotypic severity of PRPH2 associated disease. Whether the ABCA4 found in this younger sister plays any role is unknown and warrants further investigation. So in all, this family presented with phenotypic variability to a great extent, leading to 2 different kinds of diagnoses. They were all associated with a PRPH2 missense mutation that was novel and pathogenic. So this really shows the difficulty to associate genotype to phenotype in the field of genetic retina specialties. Before I conclude, I'd like to point out a couple of limitations. One being that we didn't screen all genes within these patients. We didn't perform whole exome sequencing. We only screened 285 genes because it was performed by Blueprint Genetics. So gene mutations found outside these 285 genes may be present in these patients and may affect the phenotype, and we would not be able to know. Another limitation is that we didn't survey their environmental factors, dietary habits, or tobacco use, which all can play a role in phenotype. And yeah, that's about it. If you have any questions, my email is going to be in the description below, and I'll be linking this poster at the bottom as well. And I'd like to point out that if you are looking through the figure 1 through 4, I suggest basing it off of the figure at the very bottom of this poster. You can see at the left of the references, these are normal or healthy retinal findings. And we actually took these images from the proband's 35-year-old daughter, who also carried the PNPLA6 missense mutation. So if you have any questions, please let me know. Thank you for coming to my poster session.